Hello, my name is Tobiasz. I'm from the University of Arkansas. Thank you for checking out on this research. I will present you a short story about one of the most severe diseases we have ever witnessed on roses. The causal agent is a virus that was identified and fully characterized in our lab in Arkansas in 2011 and named rosette emaravirus. Once in a plant, the virus changes the normal growth of a rose into something called a witch's broom. And symptoms of that developing disease also include rapid elongation of shoots that remain red in color for a prolonged time, leaf distortion, and shoot proliferation. And very characteristic, excessive thorniness. Rose rosette epidemic in the United States is so huge that roses are not recommended for planting in some areas, rose rosette hotspots. Nurseries suffer huge losses. When the disease is detected in their stock, all that infected material must be destroyed. So far, the disease seems to be restricted to North America, and it's being transmitted by a natural vector, an aerophyte mite named Phyllocoptes fructiphilus. Aerophyte mites are microscopic, not visible to the naked eye. That's why scanning electron microscopy is usually used to picture them. So we proved that this species transmit rose rosette. But for me, one of the most important questions from the beginning was, is Phyllocoptes fructiphilus the only vector of rose rosette? Especially having in mind that there are other morphologically very similar species on rose. For example, the key diagnostic features between fructiphilus and the other two Phyllocoptes species from rose that are presented here are just the short lines on the prodorsal shield. Those mites were never compared using molecular methods. So we decided to study that group better using combination of tools, molecular techniques and morphometry, verify their real taxonomical status, and also investigate on the possible role in rose rosette transmission. By using direct RT-PCR method developed in our lab for individual aerophyte mites, I was able to sequence over 300 populations of mites collected across United States, including major hotspots of rose rosette. So what I'm showing you here on the left is just a simplified cladogram. First of all, we have identified distinct lineages within the confirmed vector, Phyllocoptes fructiphilus, with one lineage from Nebraska being quite a distance from all the other. We don't know yet if this new lineage is capable to vector the virus, and if so, at what efficiency. But what's even more intriguing, we have found a completely new aerophyte species in the samples collected in Arkansas, Kentucky, and Michigan, and we named it Phyllocoptes arcani. Arcani meaning hidden, secret. This new species is also distinct morphologically from all the other Phyllocopteses in that group. We prepared a description of Phyllocoptes arcani, and it should be published soon. Phyllocoptes arcani shows 22% divergence in the CO1 region to fructiphilus lineage that was used in rose rosette transmission experiments. And also the other two species, Phyllocoptes adalius and Phyllocoptes rhezobius, group in separate clades, with our analysis showing that all four should be treated as separate taxons. And interestingly, the new species, Phyllocoptes arcani, was present only on rose rosette symptomatic plants. So we were wondering if it's also capable of transmission. The direct RT-PCR method that we are using in our lab allows for a detection of a viral RNA in individual aerophytes. So I have used the individuals of Adalius, fructiphilus, and arcani. I exposed them to rosette infected tissue for 3 days, and then I tested them with that new protocol. And as you can see here, rosette specific amplicons were obtained in the case of a confirmed vector, Phyllocoptes fructiphilus, but also for the new species, arcani. We have to bear in mind that the detection of a virus in a mite or an insect feeding on infected tissue, of course, does not prove or guarantee transmission. But in case of a microscopic aerophyte mites, as you can see here, it may help to narrow the list of species for transmission trials, maybe mainstreaming the vector identification process. So the new species will be used in the transmission experiments, as we are building now a clean colony of it. And if it's capable to transmit rosette, that would be the first case of an emaravirus being transmitted by two separate vector species. As we improve our understanding of Phyllocoptes species from rose, we also wanted to move forward trying to find any possible solution to combat the disease. And we are working in two areas, looking for effective biocontrol agents against the vector or the vectors. And one is the area of entomopathogens or acaropathogens, and the other one are predatory mites. This is predatory mite that usually feed on a bigger prey, like thrips or whiteflies, but many predatory species, especially from the family Phytoceidae, are known as effective biocontrol agents of aerophyte mites. Some even exhibit a preference for aerophytes as a food source. By living in the narrow spaces as well as in galls, aerophyte mites try to escape that predation. So selected predatory species are available commercially and are used to control, for example, tomato rosette mite or aerophyte mites on roses in Europe that are grown for cut flowers. So I have checked all of the samples that I have collected across the states also for predatory species. We wanted to check if some species are more often present on roses infested with fructiphilus or arcani. This was never studied before. What predators are most prevalent on the roses infested with the vector or on symptomatic plants, with the ultimate goal of selecting of 2, maybe 3 species for further field experiments. And I have recovered over 350 individuals representing at least 12 different predatory mite species. The two presented here were the most often encountered in our samples, Western predatory mite, Calendromus occidentalis, and Amblyseius andersoni. Both belong to the Phytoceidae family. Both are commercially available, with andersoni being offered in more than 50 countries. What's important is that both are classified as type 3 predatory mites, which means that they can feed on not only different kind of pests like whiteflies, thrips, aphids, aerophyte mites, spider mites, but also on various kinds of plant pollen. And this is definitely an advantage because it allows the population of the predator to develop on plants even before the pest is present, or they can survive on the plants longer after the population of the pest is low, ensuring the continuous suppression of the pest or preventing the new outbreaks. So these two species should be used in further field experiments against the vector of rose rosette. So we started from the vector. I'll talk about predatory mites, and now is in the final part of my presentation. I would also like to talk about fungal acaropathogens. One of the colonies of Phyllocoptes fructiphilus in our lab had collapsed because of some fungal infection. And as you can see here on the pictures, mite bodies have been parasitized with some fungus growing out of them. So the idea of looking for a pathogen, a caropathogen, for a vector of rose rosette has some merit because of the nature of a vector, Phyllocoptes fructiphilus. This species is a refuge-seeking type of a mite. It seeks shelter, protection around buds, inside flowers, under the scales of flower buds. And eriophyte mites, due to their size, are frequently submerged in a dew or a free water. They inhabit moist, tender areas on the plant surface, so microclimatic conditions that in theory should favor the growth of a fungus, but are hard maybe to reach with conventional pesticides. So different acaropathogens are reported in the literature, and some are used commercially. If properly selected, entomopathogenic products surpass the efficiency of conventional pesticides. So we DNA barcoded few of these fungal cases, and it turned out to be a member of the genus Meira. Meira is a group of yeast-like fungi placed within the order Exobasidiales. There are 5 species of Meira reported to date, with some proven to be associated with eriophyte mites, and some reported to have the antagonistic effect not only on mites but also on the powdery mildew. It was pretty easy to grow this fungus on different media in the lab. I harvested Blastoconidia and started experiments with spraying of mites in cages. So the Meira we used was able to kill 43% of mites after 7 days, which is less to what was observed by other authors for citrus rust mite, where mortality, as you can see here on the right, was almost 100%. But they used different Meira species and at much higher concentration. And we just started our work, and we will continue it, as having an effective acaropathogen against the vector of rose rosette could be a game changer in our efforts to control the disease. Concluding, there is no doubt for me that mites that vector rose rosette can and should be managed. We need to continue the studies of those mites and search for effective control agents in order to find a solution that could be used in the field. Also, the studies on virus resistance should be continued, as we should use any available option to mitigate the disease. By saying that, I hope you enjoyed this presentation, and please contact me if you have any questions. Thank you.